Okay, we left off on our last podcast talking about the 3 types of point mutations, an example of a disease that can result called sickle cell anemia. Now write in your notes that the next class of mutations we want to discuss are called frameshift mutations, where a shift in the reading frame actually occurs and that this shift changes everything downstream of that mutation. This can result in insertions where bases are actually added or deletions where bases are actually lost in this process. Question: where would this mutation cause the most damage? At the beginning or at the end of the gene? Now, if you said at the beginning, you'd be correct. Disrupting the code and sequencing at the beginning of the processing of a protein has a much greater chance of altering its function. So get that in your notes. Let's see how frameshift mutations work using the English language as an example. So, you see now the gene sequencing up here, the original gene sequencing. The rat and the cat ate the red bat. Now, let's look at an example of a deletion. So, if we deleted now a character, we can see now an A that's been deleted here. See the rat here. Now you can see how it shifts now this whole line of code so that it doesn't make any sense. Same thing now with an insertion. So if we have an insertion in here, we can see now, so you have 2 A's here, so rat's now 2 A's. If we have that insertion now, we can see how it changes now the code. So if we go to the triplet codes now, they're completely different, just like they are here. So write in your notes that cystic fibrosis is an inherited disease that is relatively common in the U.S. and results from a frameshift mutation. It affects in 1 in 2,500 births, and 1 in 25 Caucasians now are carriers or hybrids. Also write that cystic fibrosis affects multiple parts of the body, including the pancreas, the sweat glands, and the lungs. Let's see how this affects the lungs, for example. Now write in your notes that people without cystic fibrosis have working proteins that allow salt to enter the airspace and water follows by osmosis. The mucus layers dilute and not very sticky, which is a good thing. However, in people with cystic fibrosis, non-working cystic fibrosis proteins means no salt enters the airspace and therefore no water follows it. Now the result is the mucus layer is concentrated and very sticky, which can cause problems. People with cystic fibrosis have lung problems for 5 reasons we want to get in your notes. First, proteins for diffusion of salt into the airways don't work. This results in less diffusion. Second, less salt in the airway means that less water in the airway as well, which means less osmosis. Third, less water in the airways means the mucus layer is very sticky. We call that term viscous. Then sticky mucus cannot be easily moved to clear particles from the lungs. And as a result, sticky mucus traps bacteria and causes more lung infections. Therefore, because of less diffusion of salt and less osmosis of water, people with cystic fibrosis have too much sticky mucus in the airways of their lungs and get lots of lung infections. Therefore, they're sick a lot. Make sure you get all these symptoms and their causes in your notes. So we see now the actual mutation that occurs. You can see here that the frameshift causes the protein phenylalanine to actually be deleted in many patients with CF. That's an important point to have in your notes as well. You can also see how babies and toddlers with CF have to be percussed, meaning their caregivers have to actually strike their patients on the chest or back to loosen the thick mucus that's built up in their lungs to get them to cough it up. Okay, now that's it for our discussion of mutations. So point and frameshift are the ones that we spent the most time on. Now let's move on. You now understand that the central dogma of molecular biology explains that DNA codes for RNA, which in turn codes for proteins. You also learned about the important roles of messenger RNA, transfer RNA, and ribosomal RNA in the protein building process. But we now know that RNA does more than just build proteins. Write in your notes that RNA has many jobs in the cell, including jobs that have been traditionally associated with DNA and proteins. Let's look at some examples that have a good chance of appearing on the AP exam. Now we'll soon learn that enzymes are catalysts. Write in your note that catalysts build and break down molecules at a high rate, in fact quick enough to sustain life. Scientists used to think that all enzymes in the cell were proteins. Then it was discovered that some RNA molecules can be enzymes too. These so-called ribozymes are rare, but they play a key role in the cell. Recall that in the ribosome, RNA joins amino acids together, allowing cells to build proteins. Write in your notes that some messenger RNA molecules contain self-splicing introns, which can break and rejoin the mRNA strand. Also write that a ribozyme in the RNA ACE-P complex activates tRNA molecules by clipping off their ends. Also write that ribozymes provide further evidence that RNA may have been the first molecule of life, and that in primitive life forms, RNA may have both catalyzed chemical reactions and stored genetic information, and then had functions that were later taken over by DNA and protein. That's not been proven, but we're moving toward that kind of a theory at this point in molecular biology. Okay, now write in your notes that some RNA molecules can silence specific genes, turning off the production of proteins that are not needed at a certain place or time, and that this job is especially important during development, when cells begin to differentiate into specific types like muscle, skin, and liver. Now you already know that each cell type needs only a fraction of its total genes to be active in order to do its job. Get in your notes that gene silencing RNA molecules recognize specific gene sequences through complementary base pairing, and that these RNA molecules can shut down portions of the genome, turning off protein production. Now the way RNA does this is truly amazing. Get in your notes that RNA does this by recruiting proteins to modify histones, otherwise known as the epigenome, E-P-I-G-E-N-O-M-E, and that these modified histones wrap around DNA tightly, making it inaccessible to the transcription machinery. Man, can you imagine that? That's amazing. Now let's talk about RNA interference. This is the idea that scientists can use the cell's gene silencing machinery to study genes. Let's see how. Write in your notes that first they build a small RNA molecule with a nucleotide sequence that matches a specific gene. and that when the RNA is injected into the embryo, it finds the gene and turns it off permanently so that no protein is made from it. Now, in turn, by looking at what happens as the organism develops without the gene, scientists learn something about the gene's natural function. So make sure you get that in your notes. Also get in your notes that some RNAs silence harmful DNA sequences that sit in our genomes as relics of our evolutionary past. Now, two examples are like transposons, also otherwise known as jumping genes, write that down, and genes of infected viruses that made their way into our ancestors' DNA. So get those two things down. You see them here in the slide. Also write that RNAs inactivate viral genes in transposons, keeping them from harming us. So that's an amazing function. Got to have that. Now, there are a variety of RNA molecules that help the cell to fine-tune when, where, and how much of a particular messenger RNA molecule, and by extension, a particular protein is made. Write in your notes that regulatory RNAs can act on just about every step of the protein production process, and that some RNAs called riboregulators, write that down, riboregulators, you see it right here. Some RNAs called riboregulators bind DNA switches to turn genes on and off, while others interact directly with messenger RNA molecules to alter splicing, and therefore protecting mRNA from harm or cut it to pieces. So get those in your notes in terms of fine-tuning. It should also be no surprise to you that RNA would respond to changes in the environment. Write in your notes that ribose switches help some cells respond to external signal. Because they're so small, ribose switches are found on large mRNA molecules, and they fold into intricate shapes. Metal ions, amino acids, or nucleic acids bind to this ribose switch, which causes the shape of RNA to change. This shape change always affects whether or not the mRNA is translated into a protein. Also write in your notes that in bacteria, ribose switches regulate mRNAs that codes for proteins involved in metabolic pathways. Now, also get in your notes that scientists are building RNA molecules and using them as tools to diagnose and even treat diseases like cancer, diabetes, arthritis, heart disease, brain diseases, and viral infections. Also write that scientists can design RNA molecules that will attach to specific nucleotide sequences in a gene or mRNA molecule. These RNA molecules could someday be used to inactivate a broken disease gene. As a diagnostic tool, RNA molecules can be designed to identify certain substances in the blood that are only present with a certain disease. Write that misbehaving RNA molecules can also cause diseases such as Alzheimer's and other neurodegenerative diseases. The more we learn about RNA's role in these diseases, the more prepared we'll be to treat them. Viruses continue to astound scientists by violating the central dogma's standard rules of protein synthesis. As you know, most organisms use DNA to store genetic information, and it's this DNA that passes from parents to offspring over generations. But some viruses, including HIV, the virus that causes AIDS, use RNA to carry genetic information. Write in your notes that RNA viruses are known as retroviruses. Also write that retroviruses like HIV carry an RNA genome which is converted into DNA prior to its insertion into the human genome. Also get down that in order to carry out this conversion, the RNA must be reverse transcribed into DNA. So that's the opposite of what RNA normally does. Get in your notes that retroviruses produce a special enzyme called reverse transcriptase, which conveniently performs the process of reverse transcription. In other words, converts the RNA in the virus into DNA. Once the viral DNA is inserted into the human genome, the standard rules of central dogma then start applying again. So it violates the rules of central dogma, then takes advantage of them. So they are extremely complicated molecules. Okay, one last anomaly. Prions. Write in your notes that a prion gets its name from their function. Prion means they're proteinaceous, infectious particles. So prion is short for proteinaceous, infectious particle. And these are infectious agents that appear to have a unique method of information transfer. Prions are proteins. In other words, they don't carry a genome. As such, the standard rules of central dogma do not apply to prions. Write in your notes that prion diseases such as mad cow are thought to result from the interaction of misfolded prion proteins with normal prion proteins, and that these interactions result in the misfolding of the normal prion proteins, in other words, information transfer, and ultimately the typical spongiform encephalopathy associated with prion diseases like mad cow. That's a large mouthful. Make sure you get it all down because it has absolutely been on an AP exam. Okay, that takes care of part 2 of our Mutations podcast. Take a picture of these notes, submit them to Moodle, and pick up that study guide, answer the questions, and then let's move on. We're moving our way towards enzymes now. So I'll see you next time. Bye.